Good evening, everyone, and welcome. We are having our June first Thursday Speaker Series webinar this evening, and we're very fortunate to have Dr. Mandë Holford, and I will provide some background about her in a few moments, but welcome, welcome. And we expect a full house this evening. So I just wanted to open with a couple of things. One is, how do I know Mandë? Mandë and I met at the American Museum of Natural History around 22 years ago, and we both really went in two wild, separate directions. And but I'm very happy to be able to host her this evening as part of our series, our talk series, and we're very grateful that she has been able to make time for us. And I say that because Mandë is about to make a very huge move in her life and a new adventure, which I'll tell you about in a moment. I just wanted to remind everybody that we have some upcoming events. One of them is tomorrow. It's not our event, but it's at the New York Aquarium, and it's a World Beach Cleanup event that will also have exhibitors from various organizations right on the boardwalk. That should be interesting because it's usually very windy, and I don't know how we're going to do with the tabling part, but the beach cleanup part promises to be very exciting. And then additionally on Saturday, this coming Saturday, the very next day, at Beckzak, which is it's the urban river at Sarah Lawrence College on the Hudson River. And they are having a community event where they will be seining for fish, and it's part of the World Fish Migration Day. And so other environmental organizations up the Hudson will be performing the same activity, and somehow or another, the data will be combined. Um, and then later this month, on June 21st, Nesmia is organizing its own field trip to Big Brook State Park for a fossil collecting trip. And we hope that people in this group can join us for that. And also we'll work on providing carpooling, and it promises to be a big long day in New Jersey, seining for or sifting for fossils, and the kinds of fossils that you traditionally find in the creek in that park are shark fossil teeth and alligator jaws. So we hope you could join us for that field trip. That promises to be very exciting. So without any further ado, I'd like to provide background on Dr. Holford. Um, she is a marine chemical biologist and is dedicated to finding the next generation of venom-derived treatments. In fall 2025, she is moving her lab from the City University of New York's Hunter College to Harvard University. Woohoo! Where she will be a professor and curator of mollusks with a joint appointment at the Harvard Museum of Comparative Zoology. Harvard is building her a new aquatic lab where she will keep a collection of venomous octopuses, squid, cuttlefish, and snails. Let me know which ones I should not be eating. It will be the only facility outside of the Woods Hole Marine Biological Laboratory that creates its own genetically modified cephalopods in an effort to unravel how they produce and deploy their venom. So without any further ado, welcome, Dr. Holford.
Thank you. Thank you so much, Lisa, for the invitation. I'm excited to be with you guys today. And I'm really happy that this organization exists because I think it's important that we get science out in all these different formats, right? So speaking to scientists is one way, but also speaking with the public and especially trying to engage both learning. We used to call it learning, lifelong learning at the museum. They have a new name for it now, but it's very good that everyone is here today. And I thank you for sharing your time with me. I'm gonna start my presentation. Um, let's share. I'll put it in.
And before you start,
Hmm?
Russell, could you, okay. Yeah, great.
Is that okay?
Yeah, it's perfect.
We'll turn off the closed captions. I don't think we need them. So thank you guys. And as Lisa said, I'm going to tell you a little bit about the research that we do in the lab, but I also tell you about how we are trying to assist educators. And so bringing the power of Venom into both laboratories and classroom. And on the call tonight, I have Jessica Ochoa Hendricks, who's the CEO of Killer Snails, which is the EdTech spinoff from my lab, where we make learning games, immersive and board games for kids in grades 3 through 12. And so this talk is really about models and how models can be an inspiration. So you can unmute and tell us what you think this model is, if you know it, if you recognize it, or you can put it in the chat, although I can't see your chat.
Okay. So, well, I mean, it looks like a model of what a bird can do, or maybe a model of well, what do you call a concept model of what a a a plane or something that's
Yeah.
aerodynamic can do. Those are
Absolutely. Yeah. This is Da Vinci's model of the flying machine, which was inspiration for these guys, right? The Wright brothers, who were the first ones to get us into flight mode. And so while their inspiration was in the skies, my inspiration comes from the seas. And so tonight I'm going to take you on a journey that involves killer snails, as I affectionately call them. So here, right up, I'll start you with the exciting part of the talk. After this, you can leave if you want to. Underneath the sand is a cone snail. The thing sticking up is like a nose. It smells if there's prey in the water. The orange thing is a proboscis. On the tip, it has a harpoon filled with venom. And then instantly you see the fish is paralyzed. The snail will rise up out of the sand and have itself a meal. Whenever I'm having a bad day and I think I can't do something, I look at this video and I'm like, I can do anything because nature has shown us how a snail can eat a fish. And it probably shouldn't because you'd think the fish would get away. But clearly, the venom is so potent that it can stop the fish from getting away. So you'll see this again. This is Conus bullatus. We dug it up from the sand. It's going to inject with the proboscis, and instantly, again, the fish is
Oh, wow.
paralyzed. So the venom arsenal in these fish has several things, right? It causes paralysis. It's very instant. And what we've discovered, it's also very good at stopping pain. Because if the fish were to go into fight or flight mode, it would get away. But it is a toxin, and so dosage is important. So you'll see that the fish will recover, but too late. It's just going to get injected again and then bye bye. And so while people are very familiar perhaps with killer snails, less people are familiar with killer cephs. So cephalopods, like these venomous marine snails, also produce venom in a specialty gland that they use to subdue their prey. So this is a cuttlefish. It's going to reach out its tentacles and grab that shrimp. And then inside of its buccal mass, we can't see what it's doing. It'll inject it and then be able to digest the shrimp without it causing damage inside of its mouth. And so this is where the lab is right now. Why is this work important? It's because venom's attacks conserve physiological targets. I like to say blood, brains, and membranes, right? And so these physiological targets are the same for us as it is for the fish, as it is for the shrimp. And what we found is a conservation of how these venoms attack certain parts of these physiological targets. And so this is important because we can then use venom compounds as a tool to help us learn a lot about fundamental science. And one area in which it's been really, really progressive is helping us to figure out how the brain works. So using compounds that are found in snake venom or scorpion venom, like tetrodotoxin or bungotoxin, we've been able to map the neurons and the neural circuit. In particular, we've been able to map things at the surface of these cells, which are called ion channels and receptors, and how they are connected and how we can turn them off and on using the venom arsenal compounds. We've also figured out that venom is important because they can be used to help make medicine. And so while if I inject you with the whole venom from that cone snail, you might die because certain species are lethal to humans. But in the lab, what we do is extract individual components from that venom arsenal and use those to silence specific signals or specific ion channels, like the ones found in neurons, to prevent things like pain and cancer. And so all of these animals that you see here have produced blockbuster drugs. From the Gila monster, we have Byetta, which is used to treat diabetes. From the Brazilian lancehead snake, we have captopril, used to treat hypertension. And captopril was actually the first breakthrough drug from venom. And it not only defined a specific compound, but a whole class of drugs called angiotensin converting enzymes, which are used to treat hypertension. And then most recently, we've had from the cone snail, Prialt, which is a non-opioid acting analgesic for treating pain. And so you all are aware of the opioid crisis that we're going through because up until Prialt, most drugs on the market that are used for treating pain were opiates, and so they worked on opioid receptors, which would lead to addiction and other side effects. Cone snail drug, Prialt, works on N-type calcium channels, so it avoids the opioid receptor, so it avoids the addictive behavior. And even more recently, from the same Gila monster, we've identified Ozempic, which is being used both as a diabetic drug, but also for obesity and helping people to lose weight. So it's been proven that these compounds in the venom arsenal can, yes, perhaps kill you if we use too much, but at the right amount and with the right compound, they can also help cure you. And so venomous animals are found all throughout the tree of life. So everything red here is highlighted. And we think that there are over 200,000 venomous species on the planet that are known currently. A large majority of them are insects, but even more of them are marine organisms that we have yet to identify. And so I think now is like one of the most exciting times to be in science, not pending the current administration, but all of the stuff that's happening in terms of technology development, exposure to new surfaces, discovery of new organisms. It's really a very good time to be in science. And so why now? It's kind of because we've gone from this to this. The technological advances that we're making now allow us to do things bigger, faster, and with more accuracy than we could have ever done before. Whether it's in genomics and transcriptomics, I call it the rise of the omics, or whether it's in genetic engineering, we have lots of new tools that we didn't even have 5 or 10 years ago. And we can apply them to really complex questions, such as how venom evolved across the tree of life. How is that looking at independent traits and complex traits and looking at novelty, how these venom genes evolve in these specialty glands that we could never do before. Why now is also because of this, right? We're in a planet that is in crisis mode in terms of loss of biodiversity. At Hunter, we have a motto that says, the care of the future is mine. And I teach all the kids in my lab that we care about these organisms not because they might give us a drug, but because we want them to help sustainable ecosystems that will make our lives better if we are able to make their lives better. And so it's really important, especially as marine biodiversity researcher, that we protect the ecosystem that we're studying and learn as much as we can about their biodiversity. And so when I study venom and venomous animals, my hope is that I can turn them from agents of fear, which most people think of them as, and into agents of change and innovation. Because what you saw of a snail eating a fish, that is innovation. That is nature's innovation that allows a slow-moving snail to eat something a lot faster and quicker, which should get away, but couldn't. So um, venoms are extremely complex mixtures, right? So it's not just one thing, it's several things. They're made up of small molecules, peptides, proteins. Peptides are just smaller version of proteins. And in my lab, we mostly focus on the peptides and the proteins. And initially, I mostly worked with marine snails. And so each snail has about 250 unique peptides in its venom arsenal. The other thing to learn about venoms are that no two species have exactly the same venom. They are really species specific. So while there are common compounds that target similar molecular structures, they are not exactly the same arsenal. So what you find in a snail, you might not find in a snake, but you'll have common things there. And so my laboratory goals have been to study the venom evolution of marine mollusks using comparative phylogeny, so letting history tell us and evolution tell us how these things have evolved, identify new venom peptides in their venom arsenal, characterize the function of these peptides mostly on the surface of cells at ion channels and receptors, in the hopes of identifying compounds that might have therapeutic applications in pain and cancer. And more recently, we started to develop venom model systems because we wanted to learn more about how the venoms themselves are expressed and regulated. And hopefully that will help us make smarter therapeutics. And so tonight, that's the part I'm going to tell you because it's the most exciting part, because it's the newest part. It also means the part I know less about, but it's always fun to talk about stuff you don't know in detail and have others help you figure out, well, how you can go in the future. And so this is basically a story about our venomous path from snails to cephalopods. And so we've, for a long time, the lab has focused in investigating how venomous snails and the compounds they produce in their venom glands can be used to, as I said, either be involved in manipulating signals, whether it's a pain signal or a cancer signal, and also helping to figure out how so many versions of these marine snails have evolved over time. And so, what people don't know as much is that cephalopods also produce venom in specialty glands, and they use it both for predation and for defense, and even in sexual domination. So, you may have seen the video of the blue octopus, the blue ringed octopus, who is subduing the female with a venom TTX in order to capitulate or copulate, I said. And so in this journey that we're on, the hope is to use these compounds that are found within the venom, both for drug discovery, but also, as I said, for understanding more about how venom evolved in nature. And so, in studying the development of these venom models using marine mollusks, I'll tell you first, we look at them in a phylogenetic history. This is how we studied our snails, and we're going to transfer that to the cephalopods. We then identified different compounds in the venom arsenal, because as I said, there are several, with the hope of identifying unique compounds that are very good at manipulating ion channels and receptors. And so one such story that we focus on is identifying compounds that target tumor cells and not healthy cells. So the tumor cells are shown in pink, the healthy cells are in green. And what we're looking for, because these venom peptides are very sometimes specific to their molecular target, are things that will just target overexpressed channels in tumor cells and not target channels in the healthy cells. And in doing that, we'll be able to make a more selective drug for treating, in this case, liver cancer. And so part of the treatment of cancer is that things on the market currently are not selective, so they attack both normal and healthy and tumor cells at the same rate. But we think these venom peptides are very good at discriminating between those two, and only will attack the tumor cells. And we have one thing in the works for that. So I tell you this only to show you that we have everything we need to study and characterize the venom compounds, like using omics, proteomics, genomics, transcriptomics, bioinformatic tools, and even functional assays. We can develop natural product drugs from this arsenal, but we are missing a very valuable piece of the puzzle, and that's really the genetics behind how these venom compounds are made. And so most of the people who study venom in the past are like myself. They have a training in chemistry, not in biology or evolution. And so we tend to focus on the end products, right? So we focus on the amino acids, the peptides, the protein. But by focusing on the end products, this has led to a huge knowledge gap. And I like to describe it as kind of like a car. Like we can be very descriptive and talk about all the parts of this car, the wheel, the headlights, you know, the hood, but we really don't know how the car works. To understand how the car works, We have to go back into the factory, right? You have to go to the assembly line and understand how this car is really put together. It's kind of connecting what we can see with what we can't see. And so what we need to do to really leapfrog in venom research is to connect the phenotypic and genotypic biology that's going on. And to do that, we need, that's how we're going to be able to answer really complex questions like how did these venom genes become weaponized? The current hypothesis is they started out as regular housekeeping genes, ribosomal genes and things like that. And then they got co-opted into the specialty gland, the venom gland, and then neo-functionalized into being toxins. But no one's been able to really test that because we don't have a model system for doing that kind of genetic work. And so, as you know from childhood, models help us learn. We need models to figure out how things work. And so, there's currently no genetically tractable multicellular venom model system. My snails, as much as I love them, we have not been able to breed them in the lab. And you kind of have to collect them wild caught. So, you get what you get. You don't get upset, right? You can't say, I want an embryo, or I want three females, or I want five males. It's not possible. These are the gold standards for how science is usually done. These are the model systems. So, you have fruit flies and mice and C. elegans and people use those because they're easy to reproduce in the lab. You can get them at different developmental stages and you can really do genetic experiments. Everything we learned about genetics came out of these blockbuster gold standard model systems. But none of these are venomous, so useless to me. And so we are now having a new focus in the lab in how we're looking at how venoms develop by moving not just from studying snails, but moving to studying cephalopods. And so because cephalopods and snails are mollusks, we can do this comparative analysis of marine mollusks. So all the things that we learned from snails is not for naught. We can transfer some of that to the cephalopods, even though there are millions of years of evolution between them the venomous arsenals, we can still determine the evolutionary novelty in the gene and their venom gland tissue development. And then we compare the molecular mechanisms for regulation and expression of their venom arsenal bioactive compounds. And so it's like hitting the jackpot. Sometimes you get lucky in science. And so it was like, we can study cephalopods. And so developing venom model systems we are doing two things. We're trying to make organoids. And the reason we're making organoids is like because it started actually during the pandemic where we could not go out and collect our snails anymore or collect anything because expeditions were shut down. And the thinking was if we could just make these organoids, which are a 3D representation of your gland in a petri dish, it's kind of like evolution on the bench top then we can stockpile them like, beans and toilet paper. And we would have them always for us to be able to do our research without having to go into the field and collect all the time. So we're interested in working on venom gland organoids. And then we also wanted to use the cephalopods specifically to do genetic manipulation because all of the cephalopods that we're working with have solved genomes. So we can identify where on the specific chromosome each gene is being expressed. And then we can use genetic tools to manipulate those genes and therefore affect how the arsenals are being composed and also maybe how the gland itself is being developed. And so the last part of working with the cephalopods is only possible because of really significant technological springboards that happen in Woods Hole at the Marine Biological Laboratory, which I started going about 3.5 years ago. And they have this great bakery called the Pie in the Sky. And for me, really, it was like manna from heaven. When I went there and I learned about their cephalopod husbandry and culturing program, and it really was like Amazon, like you can call up and say, give me 5 embryos, give me 6 females, give me 5 males, and they would ship it to you FedEx and so you have it next day. And so it really broke through the way that we could think about making model systems to study venom evolution and venom function. And so now that we had all these cephalopods, which I know nothing about, we had to figure out ways in how we could manipulate their genomes to make these genetically modified organisms. And then we can develop these really new model systems for studying venom that can radically transform how we generate, manipulate, and utilize these systems. And so most people know cephalopods like this, right? So where in this coral do you think you see the octopus? You can unmute or use your pointer or whichever to identify it before it comes out. There it is. And so most people know cephalopods because they're very good at camouflage. They ink, they're speedy, they're very intelligent. They have all these different traits, but not many people have studied cephalopod venom evolution. And so that's what we're doing. But that requires me learning a lot about cephalopods. So like how the octopus teacher movie was there at MBL, I had a whole bunch of octopus teachers. So Roger Handlin, whose video you just saw of the octopus in camouflage, is one of them. Also, Carrie Albertine, who's an assistant professor there. Brett and Taylor are the two guys who figured out the husbandry of these animals and how to keep them and breed them. And so you can have them in all different life stages in a reliable way, along with Miranda, who also worked in the MBL MRC, the Marine Research Center, and then Lisa Abo is the veterinarian who's the director of the center. So it takes many hands. It's like a village, right? To be able to learn about these cephalopods and figure out how to use them effectively in research. And so what do we know about venom evolution in cephalopods? Not much. We know basically that the coleoid cephalopods, which are basically squids, cuttlefish, and octopus, all tend to have these venom glands. We know that perhaps the nautilus does not have the venom gland. And so there are a lot of questions along the way about how it may have evolved in cephalopods. Um, and so for us, we decided to stick with the squids, cuttlefish, and octopus. And most of the venom in these cephalopods are produced in what we call the posterior salivary gland. And so this is the gland that it connects between the esophagus and the buccal mass. So up here is the buccal mass, and then these two lobes down here. In octopus, there are two posterior salivary glands. Most squids only have one, and cuttlefish also have two. And so we built this in-house transcriptome database based on what we found in repository places like the NCBI, and from also what we did fresh transcriptomes from these dissected venom glands. And so we used three different squids, two different cuttlefish, and then four different octopus to make up our database. And what we found is that there are different kinds of compounds in the cephalopod toxins, and most of them are either paralytic or hemolytic. Some are coagulatory. And of course, because it's the posterior salivary gland, you also found a bunch of the digestive enzymes. And so when we identified these peptides and proteins, there's a whole bag of things which are very, very similar to what you will find in the venom from snails and snakes and scorpions. So again, it helped to solidify this story that venom is a convergent trait and that it's involved independently in all these different organisms. One of the things that are unique to cephalopods, though, is this thing called cephalotoxin, which is a very huge protein that we found mostly in cephalopods, but you don't really find in any other organism. And so when we looked at our transcriptomes, we found cephalopods in the eberrii squid. We found it in palei squid. We found it in S. bandensis cuttlefish. We found it also in S. officinalis, another cuttlefish. So we're like, oh my, this is a great marker. We can use this as a marker now to try to track development of this particular venom gene across these different taxa. And so basically what we found is that the CTX, or a cephalotoxin, is expressed mostly in squids and cuttlefish, but we didn't find any in octopus. And so what we've identified are these two unique venom gene families with CTX1 and CTX2 that's expressed mostly, again, in squids and cuttlefish and not in octopus. And we did find homologs of this in outgroups. That's the group in gray. And it supports the theory that venom peptides or venom compounds are co-opted from housekeeping genes because they're not, the gene in these outgroups are not being used for predatory or defense or reasons. They're just housekeeping genes in those organisms. So now that we had our marker, the question was, can we trace this marker through development? So when does development of the posterior salivary gland start? Do all the lineages start at the same time? And when do they start to produce these toxins? And so what you're looking at is a development chart that was made by Tessa Montague from Colombia for cuttlefish. And so you can see at stage one, it's one cell. And then as you go from stage one to stage 25, it becomes a hatchling. And so we can monitor this development and look for when exactly the salivary gland starts to develop and also when the toxin itself starts to express. And so this is a cross section of a hatchling of E. berrii, and we have sort of stained it with the CTX gene transcript for the RNA, and you can see that it's only being expressed in the two posterior salivary glands of Eberii, and it's not found in any other cross section of the animal. So this is the first time ever. This is breaking news, people. First time ever that anyone has ever visualized venom expression in a cephalopod and confirmed that the cephalopods, at least the Eberii, have it mostly in the posterior salivary gland and not in any other part of the animals. And so we're wondering, when do they actually start to produce? So having seen it in the hatchling, can we work our way backwards towards the embryos and see when they actually start to produce these venom compounds? And so this is at stage 26, 25, staining of that organism, and you can see we don't see anything there. But when we get to stage 28 or 29, you can start to see expression of the CTX in the posterior salivary gland. So that told us that it's usually a late stage expression of the toxins, but also the animal is kind of born ready. It has an arsenal and it's ready to start feeding and hunting and doing what it needs to do to survive from the minute that it's hatchling stage. And so the next thing, like I said, because we know the genome of these animals, we can then go and see where the CTX gene is being expressed, in which particular chromosome, and then we can do genetic manipulation. So CRISPR-Cas9 manipulation to try to knock out this gene. And so this is what we were attempting with palei, with the CTX2 gene. We then injected it at stage of the embryo development and wanted to see it as it grew. So this is the different embryos. This is the cryon. We removed it. And then you inject into that X basically. And then you say a little prayer and cross your fingers. And so we did this. And this is also just hot off the presses data in which the wild type of the DPLEI has two genes, CTX1 and CTX2. You can see in the wild type of the juvenile hatchling that CTX1 is expressing, but CTX2 is not. And then in the knockout gene, CTX1 is still there and CTX2 is not. So this was very confusing to us because we're like, why don't we see CTX2 in the wild type? It should be there because we know that it has both genes. But what we know is that it has both genes at the adult stage because that's what we did, right? So here you're looking at expression in the posterior salivary gland of the palei of CTX1 on top in purple and CTX2 in green. And as you go from left to right, it's just increasing in magnification and you're sort of zooming in on a particular cell. And so we knew in adults that they had CTX2 because we could see it. But what we did not know is that the juveniles did not have CTX2, and that's why the knockout didn't work. So sometimes in science, you lose. And so we're going to try again this experiment, this time injecting with guide RNA to knock out the CTX1 and not CTX2, which shows up both in the juveniles and also in adults. It also then confirms for us that, the venom arsenal changes over time. So as juveniles, most of these squids are feeding on shrimp. And then as they get older, they'll feed on different kinds of things, crabs and others. And so the expression of CTX tends to come perhaps later when the adult needs is feeding on something different and not in the earlier stages when it's feeding on the shrimp. At least this is our running hypothesis for now. And so in developing these model systems, it really helps us to investigate very new frontiers about venom, like how do venom peptides actually evolve? Is it this duplication from housekeeping genes? Is it, can we do expression, location and change in terms of upregulation? Are there other mechanisms? Can we, using genetic manipulation in places that are not expressing venom or where the genes are silent, can we turn them on so that the animal starts to express venom? And then what's driving the venom variation? Is it, neutral causes like gene flow and evolutionary divergence, or is it natural selection, like, diet, defense, and others? And then how do venoms function? Does form really dictate function? So one of the reasons we wanted to knock out the CTX gene is because it's a pretty prominent gene that's found across taxa of all cephalopods, except octopus. But if we knock out what's considered the dominant toxin, will the specialty gland develop in the same way? And how will that affect the development of the arsenal itself? Will other things become more dominant, or will they be suppressed? And so really is the form dictating the function in terms of the form of the specialty gland and the function of the venom arsenal when you start to knock out these prominent toxins. And then in terms of natural products development, once we know more about how these genes are turned off and on, it might help us make smarter drugs that are then more effective for hitting their molecular target and less leaky in terms of side effects. And so while venom fascinates me and a lot of scientists, it also fascinates a lot of kids and a lot of society. And so that's the second part of what we do in the lab in terms of trying to figure out how do we teach about venom and biodiversity in science in general to young children to get them excited or keep them excited because they're born discoverers and they're born really keyed into trying to figure out and understand questions and discover and understanding how you make them work. And so as a part of the ed tech company that spun out of our lab, which Jessica is a part of, we are trying to make science spectacular. Can you guys hear me? Yeah. And so the pain point that we're addressing is the fact that a lot of kids by middle school think that science is not for them. And a lot of teachers are looking for resources to teach science effectively using what are currently the next generation standards. And so we're hoping to hit that sweet spot by using multimodal experiential learning, where we combine immersive XR experience, which is AR or VR, with a digital science journal where the students actually are entering data and sort of our secret sauce is that all the teachers can see where the students are in the immersive experience. So teachers usually did not like having AR or VR in the room because they didn't know how to monitor where the kids are. But in all our games, there's a back-end teacher dashboard where they can actually see and monitor and track, and then be able to scaffold learning by poking the students with comments and or stickers to encourage them to keep going. And so, and Jessica, feel free to jump in if you have something to add. And so,
Just going to add on that that we also, because we know that a lot of schools don't have the hardware to support full AR or VR, there is always a web-based version. So it can all be done on a Chromebook or whatever kind of computer you may have available. Because again, not everybody has iPads or Quests or any of that kind of technology.
right. But when we combine these three components, we really think students learn and they learn to explore and discover. And then the teachers, as I said, are monitoring the live progress and offering feedback to ensure that no one's getting stuck in the technology. And so in combining again these three things, we are consulting always with teachers and we use teachers as our science advisors because we're not trying to make something that already exists. We're trying to really fill a hole. Like if it can be taught using a textbook, we're not going to replicate it in with our software. But what we're really trying to do is find a fit between teaching content and the affordances of the different platforms that we're using. And something, of course, that the kids will resonate to. And nature has all of these stories. We're just trying to present it in a way that's palatable to the students. And so one of the stories that we tell is called Biodive, and where each student takes on the part of a marine biologist, and they're going around and they're trying to figure out how plastic got into the stomach of this shark. This was done
It's
in collaboration
waterways.
with the New York Aquarium. Go ahead.
You're showing biodive. That's waterways you're talking about.
Oh,
With biodive is the one where the students are really kind of taking on Mandy's role. They're learning about how abiotic and biotic factors can impact these marine environments. And so when they're
yeah.
using the immersive pieces, they're doing it in the way that Mandy does, where they're going to a control site and a variable site. They're using the kinds of equipment that she uses to, check the salinity and the clarity of the water. And they're then using these digital science journals that you see on the page here to answer questions, take notes. And it's always been really important for us to put the students in the role of the scientist. So Mandy has always been very clear, like a lot of what we want to do is show them, like she spends a lot of time looking at computers, but she also gets to go out into the field and collect samples. So we're really trying to kind of replicate that here with BioDive.
Yeah, this is the one I'm talking about with the
Yes.
New York Aquarium
Yeah.
waterways. Because
And y'all
this
might know
one is
that
like
lady in
one
the
of our
corner.
newest one, and we did it in collaboration with the Hudson River Park and Wildlife Conservation Society. And here's where we're talking about plastics and how plastics are
Yeah.
polluting the ocean and how they're being found in the stomachs of sharks. And Marie has just been spectacular. She was the lead scientist in this one. We were basically, the kids get to track the sharks and monitor where they go over time. And then in this, they also try to figure out what are ways that we can prevent plastic from going into our oceans. And the idea is to really build stewardship for the students in understanding that the water in the ocean is similar to the water that comes out of your tap, and it's the water that you see, on the streets. And so, how do we connect all of these pieces to help students understand that they have to be a little bit more careful in thinking about how they use water?
Yeah, it's always an important part of each of these to include something specific that students can do to kind of take ownership and address some of the challenges we face because we want to leave kids feeling empowered and not discouraged. So in each of these experiences, there's like something specific that they can do.
Yes. And then our newest one is called Venom CoLab, where we're using, again, the wonder of venom to show
Mhm.
them that not only is science about a specific organism, but it's also really a team sport. And so for that, I tapped some of my colleagues, like Victoria, who works with vampire snails, Nick, who works with snakes, Meg on sea anemones, and Ashley in scorpions, to show them not only is venom a convergent trait that's evolved throughout all these different organisms, but as scientists, we learn from each other and grow by working collaboratively. So this is a collaborative AR game that we've made where the students work as team members and each student gets assigned a particular scientist role. So someone's the ecologist, someone's the molecular biologist, someone's a pharmacologist, and the last one I forgot.
Biochemist, molecular
Biochemist.
biologist.
That's
So
what I
yeah,
am. I forgot the
you forgot
last
you.
one. Anyway, and so in that game, they go through and try to figure out, looking to see can they kill or cure? What is the compounds that might be present in these venom arsenals? And in doing so, they learn about the anatomy of their different animals, whether you're a snail, a snake, a sea anemone, or a scorpion. And then they do very interactive things like this one where they're trying to milk the snails. This is actually something that some people do in the lab. You'll put the fish in front of the snail and it's going to try to inject it. And then just before it injects the fish, you'll pull it away and then it'll inject into the Eppendorf tube and you can collect and extract the venom this way without having to sacrifice the animal. And so they learn all those things. And so part of what we do again is trying to tell the story of nature in a way that's immersive, in a way that's going to spark curiosity in the students and also help teachers understand how to teach about these topics in ways that makes them informed about what they're doing and what their students are doing and helping them to work in a great way. And so right now we have free access for any teacher who's interested in trying any of these with their students in their classroom. You can just contact me or Jessica, and you can actually try out all of our games. We have tabletop games. That's how we started. Assassins of the Sea was our first game in collaboration with the American Museum of Natural History. Biome Builder is a fantastic game for teaching kids about the food chain and learning about biodiversity in different biomes. And like I mentioned, Scuba Adventure, no, Biodive, Venom CoLab, and Waterways are also the big immersive games that we've made so far. And so what we really are trying to do is elevate science in the classroom. And from our feedback, it's been fantastic because the number one goal was to try to attract and retain middle school, particularly middle school girls in STEM. And all of the products that we've tried and we've tested nationwide, all of the products that I've told you about today, and we always get a feedback of close to 100% of the students saying that, they've learned something new, they're excited, and they want to continue to learn more, which is the ultimate best thing ever. And so this is our team of killer snails that Jessica, that's Jess in the red, leads. And I
Oh.
basically just come in and talk about the science. So that's why I don't always remember and get things right because I'm just making sure the science is accurate and Jess is doing all of the other work. So I hope that this very journey from snails to cephalopod has made you realize that, oceans are the future and all that we need to learn and all that we can do. And we must protect our oceans and the creatures that live in them. And so while these models are inspiration, I thank all the people in the lab because, you never do hard things alone. And so it's all a big collaboration between the folks in my lab and then Jessica and Killer Snails and then the folks in MBL and all the other collaborators that we work with. And I thank you very much for your time. And again, if you're interested in using any of the products, we're happy to give you free access so you can try them in lab. And we don't bite, only the mollusks do. So thank you again for coming and spending your time with us. I'm gonna, yay,
Thank you so
great
much. What a
question.
great presentation. Really very informative and exciting. And you both have done quite a bit of work. And I think what we could do right now is open it up to Q&A and see what questions our visiting audience may have. So if anybody wants to drop it in the chat, or if they want to just ask directly, we could do it that way as well. So I don't mind starting, get things rolling a little bit. And I just want to say that when you got up to the part about what's the stage that the venom is produced in the salivary gland, I thought that was really interesting because right away I started thinking about what your next statement was, which is at what point do young cephalopods start hunting and then using that newly formed venom to be able to catch food, and then the other point you made right after that was that the venom changes over time from juvenile to older individual. And so how does that translate into what they're actually going after?
Yeah, that's fascinating. We're starting to map that out now, but we know from like snakes and snails and scorpions that their venom does change over time. When you collect the juvenile and you do transcriptome or proteome of the compounds in the venom gland, it's very different sometimes from what you'll find in a full adult snake, scorpion, or spider. And so having seen that now also in cephalopods makes us think that this is a feature of the venom arsenals across different species.
And before stage 28, let's say, what are the cephs actually eating? Where are they getting their nutrition from if they're not poisoning other animals?
Yeah, no, at that stage they're just like little embryos developing, and it must be, I don't know if they're filter feeding like some of the other mollusks do, but it's basically we're not doing anything to them at that stage, or the folks that are husbandry. You're waiting for them to become hatchlings, and then they start feeding them the shrimp and all the other stuff that they know they need to eat to survive.
Wow, how interesting. I never really thought about any of that in what I know about cephalopods. I never thought about them eating what they're eating, and
I know.
except for cone snails, because those are the ones that I knew about, and
Right,
I've seen video like the video you were showing us this evening.
right.
So so that's very informative. How about are there any questions from our audience? Anything someone would like to ask?
Oh,
Yes.
I can't find it, but yeah.
Okay, I'll go next.
Uh,
Go ahead.
okay, go ahead, Suzette, and then Meryl.
Oh, okay. All right, thank you. First, Dr. Holford, thank you so much. That was really intriguing.
Thank
And you
you.
already answered one of my questions, which was how can I get access to these games to try out in my classroom? So I can email you later about that.
Yeah, and Jessica, can you put our emails in the chat?
Uh,
Yeah.
Yeah, that would be
And my
great. Thank you.
second question had to do with in your very busy life, I'm sure you're extremely busy. Do you have time to talk to students?
Oh yeah, I always make time to speak to students. I go to classrooms and I've gone to there was the first New York City Student Symposium that was held here at Hunter High School a couple of months ago. So I
Mhm.
spoke there. I think it's important because we need them. We need students to go into STEM. And so I'm always eager to speak to them and also to have them in the lab. We routinely have over the summer some high school students come through and we have undergrads also that work in the lab too.
All right, excellent. So I'll be in touch about both those things.
Great.
Thank you so much.
Meryl,
Yeah, hi, can you hear me?
we
Yes.
could hear you. We can't see
Okay.
you, but that's okay.
Okay, yeah, okay. Yeah, I researched a little bit about the pre-alt because it's
Mm-hmm.
such a, fascinating new drug that is now available, with the benefits of non-addictive, properties. Um, but I was reading that it had such severe, almost psychotropic, psychedelic side effects that the medical society doesn't really like to use it. It even could cause someone to be suicidal. So while it looks so promising, it has very negative potential side effects.
That was a study that was done with very small group, and yes, each person that takes the medication, if it has different side effects for, and for this these particular, I think there were like three people who had a very not conforming reaction to the drug.
Oh, it's a low. Oh,
It was a
uh,
low study number, and the biggest drawback though with pre it's not all wine and roses is that it doesn't cross the blood-brain barrier, so you have to take a spinal tap for it to be delivered. And so
yeah, yeah, it's
that's the reason that it's
epidural.
mostly not
Yeah.
as widely used and prescribed.
Mm,
Right now, they only use it for patients who have lost the tolerance to morphine or, are in extreme pain because it is 1,000 times more effective than morphine. And so
mm.
most doctors want to use it, but the delivery method is a spinal tap, which not many people are going to sign up for.
Yeah,
And so it's
yeah,
not as widely used as
yeah.
possible.
Here's a little tidbit. Oh, I don't know, maybe 35 years ago when I was a volunteer reader for Sea Grant, and I was looking through piles of research proposals, and there was one about the killer snail, the cone. And I was so fascinated by it, I gave it all my points, and that was at Ithaca University. Ithaca College. And
Great.
so, yeah, so I've been following this for about, I don't know, over 30, 40 years.
Yeah.
So, anyway, I want to thank you for your brilliance and fascinating presentation. Keep up the great work. We're so proud of you. And
Thank
I hope that
you.
you remain part of the Nesmia family, so to speak, for future presentations at conferences.
Absolutely. Whenever Lisa invites me.
Okay, thank you very, very much. Very impressive work.
Thank you.
Yeah, I think there's great opportunity for both of you to present at our conference, and we had a great one at this past conference at SUNY Maritime. Thank you,
There, I was there.
thank you so much.
Did I
And we
to get
Yeah, so I
trouble?
mean, this definitely should be something that we will be considering for the next conference, and hopefully both of you can make the time because the two different areas that you're talking about, I mean, even though they combine again, but through research and education are so much in demand, and I would think that our teacher membership, our formal and informal members can benefit from a workshop. Certainly everyone's benefiting this evening from your talk, but I think it could go further, and I would definitely want to talk with you about using some of your products for our teachers and making them available in ways that are possible.
Lisa Morella had a question.
Go ahead.
Yeah, thank you. I have a kind of, I don't know, question or reflection or thank you for everything. Thank you, Miss Lisa. I'm so glad to see you again after a month. And Miss Mandë Holford and Jessica, as you were presenting, I started to make connection. Oh, I wish I knew this earlier. I wish I knew this earlier because as Miss Lisa know, my school, I teach at the International High School for Health Sciences. We are part of a an organization called
Maybe we
where we try to incorporate ocean literacy principles. And the fact that you make the connection between the venom use in the medicine goes correctly with one of the principles that ocean and human are so interchangeably connected.
Right.
The fact that you mentioned that some of these venoms have been used to treat tumors, I said, oh my God, I just finished the cancer unit with my students. So I am a little bit behind, but I was able to take a few screenshots if I'm not violating the
No, totally fine.
copyright. One concern I have is that when I took a screenshot it says available for purchase. So in this, the Killer Whale website is kind of a website that we need to have an account and to pay, or it's something that the kids can play under the supervision of the teacher. So kind of it's a free website, or the school has to have an account. And
Yeah, no, that's a great question. In general, it does cost money, but because we are huge fans of Next Man, we give teachers who attend this free access. So we have, is your school part of the NYC DOE?
yeah, I work for the
Yeah.
and my
So
idea,
we've gone through the IRMA process. So all you have to do is request it, have your principal request it, and then it'll be approved. And then you can use the login through Clever, and your students will be able to log in and access it. Email me and I'll send you all of the seats so that you have the licenses for free.
thank you. Because now we finished it. We're going to have the regions and others. So in high
In
school,
their semester.
Yeah.
it's over now. But in September, I'm going to have a huge project in ecology and ocean as well. So that's why I took your emails. I emailed
Yeah.
you.
Yeah.
And I would recommend having your principal reach out now, just because the NYCDOE, when they have to approve these kinds of products, it can take them weeks, if not months. So reach out now, and the licenses will still be good all the way until June of 2026. Just if you start the process sooner, then
So he just
you'll know.
say that one of my teachers attended this. So, and then we would need to use.
I'll send the seats to you. And then when you try and access it, it'll say your administrator needs to ask the NYC DOE for permission to connect with this site. And that's when they'll have to send that form.
And
But
then
I can send you that. Yeah.
all right. Thank you. Thank you so much.
Absolutely.
Thank you.
Yeah.
Lisa, so
go
I
ahead.
want to thank you very much. It was really a great presentation. I think some of us are going to have to watch the video and go over it again to understand everything that was going on. But a couple of the shots you showed the kids looking into VR instrument. Now, are those iPhones that are inside a box, or are
That was the
those
Google Cardboard.
specific VR instruments?
That was the Google Cardboard.
Okay.
Those pictures were actually from like 2020, back when people were still willing to share phones and share
Right.
headsets.
Before the pandemic.
Yeah. Now like I mentioned, we have it all accessible online. You can still use it with a Google Cardboard if you happen to have one around.
Right.
And you can also use it on Quest, but most schools don't have access to that.
Right.
And so you can use it all through WebVR. And that way it just opens up in browser. It's not as immersive, but you still get the first person kind of feel because you're able to look up, look down, make things bigger, scale them up, scale them back. So there is still that aspect. And again, if you happen to have a Google Cardboard, then yes, you
Yeah,
can have the app.
it's really funny. The one I got was free from the Times.
Yeah.
Probably 15 years
Yes.
ago or something like
Yep.
that, and I still
Yep.
have it sitting here. And every once in a while I come across some application that it works on,
hmm
it's a great instrument that costs almost nothing,
Yep.
except for the iPhone. So, but I just want to thank you. It really was a really great presentation, Mandy. Thank you.
Thank
And
you.
so we're going to send this video, the copy of the video, to all the people that registered. And Mandy has everybody's emails. And
Yes, you shared the list of attendees with us.
with you, right? And this presentation will be available on our website for NISMIA members. So
Oh, wonderful. Thank you.
Yeah.
thank you.
Yeah, and I want to echo Lou's words. The presentation was brilliant. It was amazing at breakneck speed. I'm going to have to review it myself.
Right.
But
And I'm definitely sending it to my two college grandsons for
Oh,
their
great.
review, for them to look at.
And I also want to thank Lou for putting together all of the promotional material and the flyer, and also Russell for facilitating the session as he always does. Amazing work, everyone. But I have to say, I'm still thinking about all the applications of this particular talk this evening, and I know I'll be in touch with you, and maybe we could brainstorm with both of you and figure out how to make this go even further. I mean, you already said, that you're opening it up free, but I'm thinking even beyond this moment in time, how we can continue to benefit from this resource that you presented this evening, because that's what we are about here at NISMIA.
Great.
So I want to thank everybody for coming. You've been a great audience this evening, great questions. And if there aren't any further questions, we'll say goodnight to everybody and see you all next month. We have a speaker from Farmingdale State College, Dr. Peter Park, who just joined our board. And I also saw another board member on this evening, Lilith Genovese from Sea Grant. So we were, everybody, we were in great company this evening. And I thank you very much again, Dr. Holford and Jessica Ochoa. So good night, everyone. And I hope to see you all again next month.
Yeah. Have a great night. Thank you guys
Thank
for
you so
coming.
much. Really appreciate it.
Bye. Thank you.